Okay, students, we're going to be talking here about some of the symptoms of diseases. And if we look at this particular picture here, I just wanted to show you some actual pictures rather than black and white. These are leaf spots. So this is an actual symptom. And another symptom that you might see on a tree would be something like this here. And this is a canker on a tree that's probably developed. The tree's trying to heal itself, so it either got hit. It's up a little high for a mower to hit it, but something hit it in that spot. I can't tell how far up off the ground it is. It might be a foot or so, but that's just the tree trying to heal itself, so that's an abiotic factor as well. And then one of the other symptoms that you might see here on this particular picture, this is a symptom of wilt, as you can see from the title down here. And it's just the whole plant is just losing turgor. And I'm trying to get that thing to stop, and it won't. So we'll stop with that. Then the next thing I wanted to show you was some of the signs. Okay, and here, I'll try to get that to focus a little bit. And that is powdery mildew, so that's a sign of a disease. Sorry, I just got a text message from the dean. So I'm going to have a little bit of a gap here until I finish with this. Okay, so there's powdery mildew, and we can see by looking at it here, here's the sign of the fungus. It keeps going in and out, and I apologize for that. Okay, I'm going to try to get this to stop here. This particular sign is, this is actually caused by a bacteria, and the bacteria is actually clogging up the xylem and phloem, and this is called bacterial ooze, and it's produced by that bacteria within the plant. So it just gums up the whole plant, so it can't get nutrients or water up and down the plant. So that's kind of cool too. And then the last thing I wanted to show you, this is actually pictures. I'll see if I can get that to stop again. Pictures of nematodes on a root hair. And you can see these nematodes are very small. With this being a root hair, they're very see-through. And actually, when you look at these on higher power, like I did, I actually went to the Netherlands and studied nematodes for 2 weeks under microscope. And you can actually tell what they are by their structures, which if this thing would stop, I could get it to, but it's actually a pretty cool picture. There we go. All right, so we've looked at both some symptoms and signs. Okay, there aren't any signs in abiotic factors because there won't be anything to look for. Okay, and I'm not focusing on this right now. Again, if we look back over what we were talking about, oh, the camera fell off. Sorry, guys, that's a pretty bad thing. All right, there we go. I am recording lecture, yeah. How'd you get, was the door open? Yes. Okay. That's my best buddy. We just had one of the students walk in. So with the disease triangle, you're looking at the plant, the pathogen being present, and then the environment. Okay, I only have about 10 more minutes. And let me see. One thing I'd like to show you is just the infection mechanism. And if we look at that, here's powdery mildew on a plant. And you can see that it's actually affecting the leaf surface. So it actually, the Haustoria actually will puncture through into the into the actual stomata of the leaf surface. So they can actually, then their conidia that grow up out of here and release their spores actually grow out of that. And the chasmathecium is where we have our spores being produced as well. So just a general thing. So I'm going to stop with that part there, but I want to go back and talk about fungi, bacteria, and nematodes. I don't know whether I can, I think I can, I don't know if I can pause or not. I better not. I better keep going here. Let me just finish with this. Okay, so the major groups, and I'm just showing this for something to show, are fungi and bacteria and nematodes. And what you want to know about fungi is that they are actually reproduced by thread-like filaments called hyphae. And then when you got large amounts or large masses of these hyphae, they're called mycelium. And that's that fuzzy-looking growth that I showed you a picture of that was on the surface of the powdery mildew. And you usually have lots and lots and lots of spores. Okay, and in your book on page 25, it actually shows you fungal development. It starts with a spore. Your spore germinates. It starts producing hyphae. The hyphae start growing. And then when you get a mass of hyphae, they're called mycelium. Okay, and then after we get these mycelium, they mature and they actually will produce a fruiting body. And then by whatever fruiting body that spore produces is what that fungal species is determined to be. So that's how they determine fungi groups or species, is by fruiting bodies. Okay. And a lot of the fungi have a single host, but there's a lot of fungi that can affect many different hosts. So you want to make sure that you know what you're working with. And again, you might have an alternate host. So like rust diseases, they have different plant species. You'll have a main host and then an alternate host. Just trying to think, like cedar apple scab. The apple requires a cedar tree for an alternate host so the rust can develop on that particular species. Okay, so bacteria are single-celled organisms. They're microscopic, and they just reproduce by simple cell division. And their saprophytic bacteria are really important in the breakdown of organic matter. So we've got a lot of bacteria that are also pathogenic to plants. And the picture I showed you earlier was of some bacterial ooze in the xylem and phloem of the plant, so it was keeping nutrients and water from going up and down, so it actually just blocked everything up and killed the plant. Okay, we have not as many bacterial diseases in Washington. We have a lot more fungi. I would say 95% of the problems we have are fungal diseases. Okay, water is the most important thing in the spread of bacterial diseases because the bacteria just spreads all over the place. If it's not wet, they die. So you're going to see my bacterial diseases are soft rots, leaf spots, wilts, blights. So this is really important in our vegetable crops and probably our flower bulbs, hyacinths, soft rotted flower bulbs, bacterial blight of Dutch irises. One of the worst rots is obviously in potatoes. So you've got a bacterial wilt of alfalfa and a ring spot of potato. Those are both very important in our crops. Notice that fire blight is also, that's a disease of apples and pears, and that's a bacterial blight. Usually you can see a shepherd's crook or a bent branch. And then this is all maples and lilacs and cherry trees get Pseudomonas bacterial diseases, which actually gums up the inner workings. Okay, viruses are even smaller than my bacteria, so they can only be seen with an electron microscope. And they are composed of nucleic acids, and they have a protein coat. So they only multiply within the living cells of their host. So viruses are introduced into host plants. We talked about this earlier about insects being vectors for diseases, so they transmit the diseases. The virus infections in plants are systemic, and they are usually a significant problem in vegetatively propagated plants, such as nursery stock and seed. So we have to be careful that we don't get the virus in the vegetative material. Tulip breaking virus is a disease affecting tulip cultivars. Curly top, transmitted by leafhoppers, occurs in ornamental gardens. One of the viruses that actually infect some of those parrot-type tulip bulbs that have the teeth on the edges of them. When you see like a red tulip with white stripes through it, that's a virus that causes, actually almost a mosaic, I think is what it's called, that actually causes the coloration in the tulips, and that actually makes them more valuable. And actually in very old world Europe, in the Netherlands and in that area, they actually used tulip bulbs for their their monetary system. They were worth so much that people would actually use the bulbs for their trading. So we can see leaf and fruit mosaic, stunting, and distortion come along with the viral diseases. And then nematodes, I just showed you a picture of that. They're just colorless roundworms. They're more closely related to parasitic worms found in some of our animals. They're in soil, fresh water, they're everywhere. But they're so small you can only see them with a microscope. Most of our plant parasitic nematodes possess a stylet, so it's got a structure, a spear-like structure in its head so it can pierce the plant. The plant parasitic nematodes are very small within the world of nematodes, and when they were classified, that's not really something that they thought about. They were kind of an afterthought. So our interest in nematodes as far as in plants are very small with nematologists. But notice all plant nematodes reproduce by eggs, and eggs are produced by a female. And the number of I can't get that out. This is on page 26. The number of generations per year depends on the soil temperature and the pH. Because nematodes are more common in warmer soils. They do shed their skin and they molt 4 times before they mature. And notice that they usually stay in their same shape most of their lives, but some of the ones that change are the female root knot and cyst nematodes swell and become lemon or pear-shaped. And then the nematode eggs hatch only when they're stimulated to do so by root excretions from susceptible plants. So it's going to stay dormant until it actually can sense those excretions from a plant, and then it'll actually the eggs will hatch, so they know there's a host there. So these plant nematodes migrate freely over the root surface and usually feed on the outer cells. There are so many different types of nematodes. Some move quickly, some don't, some require more water than others. The main thing is that they're going to require some moisture, and they don't like bright light. So the highest concentration of nematodes is going to be generally in and around the roots of susceptible plants. If you see plants that are stunted, off color, might have a damaged root system, then you might want to check for nematodes, because it spreads very slowly. The movement of infested soil or plant material is going to, if we move them around, we can disseminate the nematodes much more quickly, and also splashing of water. There are also beneficial nematodes as well, which we've used to take out other larvae and things like that. Okay, as far as dissemination, we talked about the disease triangle, and if we look at the disease cycle, they develop in 3 stages: inoculation, penetration, and infection. Okay, so if we let it get too far beyond inoculation is really where we want to be. If we get into infection, we've missed that, especially with a contact. So we've got infection here, and it's going to be, say the leaf falls off, then the disease gets spread. It produces spores from the leaf surface itself. The leaf is moving around. That spore comes over and inoculates a new leaf. It starts growing on the outside, and then as the mycelium may be growing on the top, then it'll produce a runner that'll go into the leaf, which is a haustoria, and that's what you see here, and that's where the infection takes place and actually damages the plant. Okay, and on page 27, you have a table that gives you the mode of dissemination by fungi, bacteria, viruses, and nematodes. And fungi, almost all of them are disseminated by water. The only way is that isn't is with viruses. Fungi are spread by soil, equipment, and older plant materials, seed, and insects. And same thing with bacteria. So inoculum is any part of that would infect the plant. Penetration is when the infection enters the plant, and then infection actually is where the pathogen incubates and grows and reproduces within the plant. Okay, but just because, notice this last broad factor here, or the bold print here, just because a pathogen penetrates a plant doesn't mean a disease will develop. We can actually stop it at that point, perhaps the environmental change or something like that, and it won't allow infection. So it all depends on what you have as far as the plant. And the plant populations, the last thing I'll close with today is that the plant populations are influenced by the host range, the ability to survive. In the absence of a susceptible host, some can live for 30 years without anything around. Relative susceptibility of the plant varieties, how you're managing them, your climatic conditions, and then how the pathogen is disseminated, and if you've got any vectors involved in actually spreading the disease. So you have to monitor all these factors to see how serious it'll be. So I think that's where I'm going to stop with this lecture. I appreciate your attention, and remember, we are going to have a quiz at the end of the chapter on Monday. Those of you online, go ahead and you can go ahead and fill out the quiz. And those of you in class will take the quiz in class. Thanks.